All right, we're now recording. Hello, everyone. Thank you for joining today's DoD Environmental Planning and Conservation webinar series presentation. As a reminder, today's presentation is being recorded and then posted on the DENX Natural Resources website under webinars. Today's presenter is Matthew Allender, Clinical Associate Professor and Director of the Wildlife Epidemiology Lab at U of I-Urbana, Clinical Veterinarian with the Chicago Zoological Society. Dr. Allender will be joined by Robert Lovich and Christopher Petersen, who need no introduction if you're familiar with DoD Park. They will be discussing ophidiomycosis on military lands, part 3. Following the presentation, we will have time for questions. Before we get started, please once again take a moment to make sure that your phones and web browsers are on mute. With that, I will turn it over to Matt, Chris, and Rob.
All right, thanks everybody. So I'm going to take the first part of this presentation, but you're going to get to or have to hear from all three of us, depending on how you see it. So this first part is going to be a little bit about general knowledge of where the state of the knowledge is for ophidiomyces. I see a lot of familiar names on the participant list, so many of you guys have contributed to a lot of these samples. So ophidiomyces is a fungus in the order Onaginalis. It's close to all of these other reptile pathogens that you see here, Nanisiopsis, Parananisiopsis, which those of you that are working in the Northwest are seeing a lot more Parananisiopsis in wild populations. Just like with all of the other bacterial and fungal taxonomy, the names have changed over time. But the organism hasn't, but the names have changed. So initially it was the CANV or Chrysosporium anamorph of Nanisiopsis varezi. That's now been split into all of these other three. Initially, ophidiomyces was called Chrysosporium ophidicola. And then most recently, this O was actually a typo in the initial publication. And it was always ophidicola, not ophidicola. So you will see lots of different names. It's all the same thing. These three are different. The Amidomyces we're not going to really talk about, but it's the new turtle shell fungal disease. And some of you have actually participated for Western pond turtle surveillance looking at Amidomyces. They're all related. Amidomyces, the turtle one, has a lot of diversity. Ophidiomyces, which we're talking about here, has very little diversity among the isolates. Ophidiomyces was first identified and named Chrysosporium ophidicola using this black rat snake in, I don't know, about 14 or 15 years ago, 2009. With this granuloma on the chin. The rest of the body didn't look all that abnormal. That somewhat started a wave of investigations, and then both prospective, which we're going to discuss later today as part of the DoD project, but also retrospective. And they went back into Florida for this pygmy rattlesnake population and found skin lesions that were very consistent with ophidiomyces that have now been linked to ophidiomyces dating back into the early 1980s from Florida at least. We've done a lot of work historically. This pathogen can grow at all kinds of temperatures. 25 C is the best, but if you notice, it'll grow all the way from 14 to 35, which in North America is the entire activity range of every snake that we have in North America. So it can grow, and at least from a temperature perspective, can impact every species that we work with. Initially, we just found it associated with lesions, but we didn't know if it could actually cause disease and if it needed a break in the skin or if it needed to be in severely affected animals. So we did a challenge study in healthy adult reproductive cottonmouths and showed that just the topical application of this fungus can cause disease that is consistent with ophidiomyces. This was also found in corn snakes by the National Wildlife Health Center. They needed to abrade the skin, in this case with sandpaper, in order to cause those lesions. But again, in healthy snakes with a break in the skin, ophidiomyces can lead to disease. But not quickly. Ophidiomyces, just like with a lot of fungal diseases and with all reptile diseases, go slow. From the point of inoculation at day 0 to when you start to see it in a snake, the median, so the middle, is around day 40. But some snakes, even up to 85 days, that were topically applied with this fungus, still weren't developing clinical signs at day 80 to 90. So it can happen, and this was under controlled settings. In research, I mean, in natural settings, this could be 1 to 2 years after exposure, these animals may start to show signs. So how do we tell what's going on and if they've got it? Those of you that were part of the project knew about physical exams and identifying clinical lesions, so I thought we'd go through some of those a little bit better. But the first sign usually that we see is a displaced or thickened scale. So this is just a single displaced scale, and this animal is positive for ophidiomyces, no other clinical lesions whatsoever. This tends to die and become black, which we see as necrosis. You can see it here. This one from A has moved into a necrotic cell. This is a dead scale, but it's still attached by all of the living scales that are around it. But it starts to spread, and it can form either a granuloma, which is this elevation with pustule type material underneath it. This is a Lake Erie water snake with it on the side of its head, or it can progress to these collection of necrotic scales, these scabs or these crusts that are more than one scale in size. All of this tends to lead when the dead things fall off, or when these granulomas rupture, to these ulcers. You see this red cell or red tissue underneath, that's muscle. Here it's starting to scab over, but there's a definite gap or a defect that's causing this ulcer. So they can look a lot of different ways. How do you diagnose it? Physical exam gives us our first clue, but qPCR, which is the most common and the sensitive, and what we used for this project, it's swabs or tissue of that. Obviously, for our project, we use swabs. Culture can be done, but culture is terrible, honestly. And if we had 10 known positive snakes that were having an infection, only 1 to 2 out of those snakes would actually culture positive, largely because a lot of other things outgrow ophidiomyces. In a culture, it takes 2 weeks for ophidiomyces to grow, and before that point, usually Asper or something else that is ubiquitous in the environment tends to outgrow it and kill the ophidiomyces. Biopsies, which are invasive, usually require surgery. There are no blood tests for detection of this, and what you can see here in the background is UV light. We did a study looking at Woods lamp and UV light in the detection of ophidiomyces. All of these things where light is shining, or it's shining, reflecting, is where keratin has been disrupted by ophidiomyces in this Lake Erie water snake. Back to the swabs, because it is the most common. It doesn't come without its issues. Tissue, this is a fungus that's tissue and fungus that it invades. So once it invades, it's actually not on the surface of the skin for us to sample with simple swabbing. It really takes a more aggressive swab, and you can see this as we're pulling off this scab. And scabs and these crusts and lesions are only one area, and if it's before you know what's going on, swabs in other areas of the body can be really difficult. So we looked at, prior to this, the DoD project, swabs of clinical animals have nearly 8 times higher detection than swabs of healthy skin, but the false negative rate is quite high. But between biopsies and swabs, we're really good. So we don't necessarily need to get invasive, but swabs can be under-detected unless we swab multiple times and across multiple places on the body. So a single swab on the left side even a whole body swab has a 70% false negative rate. In order to lower that to less than 10%, so to have a 90% specificity and sensitivity, we really need to swab 8 times on the body or more. And that's why many of you guys had that as an instruction on your sample collection. It was based on that previous data. And Ophidiomyces, not to get any more complicated, but does, has multiple different categories of diagnosis. So the easy one is negative. If they don't have any physical exam abnormalities and the qPCR is negative, then they're negative. Ophidiomyces present is animals that have a positive qPCR, but no lesions on physical exam. And many of you that participated in this study saw that Ophidiomyces present category where you guys failed to find any clinical signs, but those animals had the DNA present. Possible ophidiomycosis, this happened less frequently in phase 3 or the second major part of our project versus the first one. And this is in large part because everybody got a little bit better and we got better at explaining what these clinical signs were. But possible ophidiomycosis is when there are signs, but that the qPCR is negative. Most often at this point in time, let's go back. Most often at this point in time, this is because the skin lesions are caused by a different fungus. And then apparent ophidiomycosis, this is all we got to in our study because we didn't do invasive biopsies, is that you guys saw signs and we found PCR positive lesions. This is a positive sample, ophidiomycosis. You're not supposed to be able to read this, but this describes the previous study in that the taxonomic diversity of all the positives or all the snakes we tested here are on this taxonomic grid. Everything that was positive is in orange and in red, and you can see that every taxon pretty much has an orange or a red associated with it. The gray is associated with negatives, so there's also negatives just about everywhere. So it's not restricted to a certain taxa. And this is what we've been seeing across North America and other studies, is that every species of snake is susceptible. And currently there's more than 30 that have been found. So this has progressed over time. In 2008, it was really just found in Illinois and New Hampshire, and this was largely because of sampling bias. Then by 2013, it's expanded to our detection across a lot of Eastern North America. In 2016, now it's starting to go south and towards the southwest. 2018, it's pretty much everywhere we've been looking. And then in 2020, when we did our first DoD investigation, we identified it in Idaho for the first time, Oklahoma, and Puerto Rico, in addition to lots of other states. Gray is negative results. White, we didn't sample in that study. Also, since this period of time, we've also detected it in California. So it's coast to coast entirely. It's a complicated slide from the very first DoD project, but pretty much look at the black and the gray on these boxes. And these are for each of the individual bases that we sampled. And you can see that there's a lot of diversity. The black is the apparent ophidiomycosis, so the animals looked bad and they were positive. This is really restricted towards the East Coast, Eastern North America. The new states really had detection of ophidiomyces, but not necessarily associated with clinical signs quite yet. So even though there's not a lot of genetic diversity or different species of ophidiomyces within ophidiomyces, we are finding now 6 new clades. There's a European clade, and there is a clade that's been really associated with captive animals, but then the other 4 are associated with North American species. Clade 2 is most common, but it can be found just about everywhere. This wasn't obviously part of our DoD, but often something I get lots of questions about. There are treatments that are available. This is a treatment we developed especially for venomous species where we can nebulize terbinafine into a 10-gallon aquarium, and these animals can then absorb it by breathing it in, but also on their skin. And it's been really effective for certain species, but not for others. All right. Disinfectants, this is for everybody here. We did a disinfectant study. The number of colonies that grew is listed here in the table. So you can see lots of things failed to allow growth of Ophidiomyces, bleach, at various different concentrations, ethanol, a lot of the benzyl products, but things that allowed growth. Simple green, don't use it. Novosan, which is chlorhexidine, which is something we commonly use in the veterinary profession, doesn't work. And interestingly enough, this was one of our biologists asked to do me to test some of those broadcast antifungals that are used for bushes and fruit trees. And as you can see, this grew better than the saline controls. And these azoles were first approved by the EPA for use in the mid to late 90s. And we saw a switch in Ophidiomyces shortly after that. But these do not work. We did a challenge study looking at the effect of temperature, and temperature does have an effect. That the survival time is longer at lower temperatures than it is at higher temperatures. But the distribution of lesions and signs and those types of things didn't really impact it whatsoever. And this is a really, these last two slides that I have are really just for fun. But one of the challenge studies, we looked at both control and treatment animals at two different temperatures for prairie rattlesnakes. And we looked at what genes were being expressed in the liver, kidney, and skin. And what we can see is that the cold treated animals really isolated in the liver and the kidney and the skin differently than the warm treated or the control animals. And the cold treatment animals had 100% mortality, whereas the warm treatment animals only had a 50% mortality. And there were differences in upregulation of lots of genes in the liver and the kidney, and downregulation in the liver and the kidney, basically to tell us that what's happening, and one of the things we didn't know is why animals are dying from snake fungal disease, because they really just had skin lesions. And it's like, this shouldn't be killing these animals. They're not amphibians that are having, to utilize their skin for numerous different functions. These really just have protective functions. But what it turns out is that even in the skin lesions, these animals are upregulating and downregulating genes in their liver and kidney that indicate that these guys are having complete organ shutdown with and kidney and liver failure with snake fungal disease. So anyway, that's why we care. That's some of the current state of the literature. Now I'm going to pass it off to Chris to kind of talk about this project and the scope of snakes on DoD lands. All right, Chris, take it away.
Thank you very much, Matt, and we really appreciate that background information. Hey everyone, this is Chris Petersen, and the next couple slides are going to be about what we know about snake diversity on DoD lands. And while I'm going through these slides, you're probably wondering, well, how did they obtain all this information? Well, all of what I'll be presenting on today comes from a very extensive inventory that we did for reptiles and amphibians on DoD lands, which took us about 4 years to do, but we inventoried over 400 military installations and determined what species were confirmed present on those installations, which ones had the potential to be present, and then formed a database, which we still maintain even today. And that database gets maintained and updated almost weekly. So it's an incredible resource for us for looking at the diversity of herps on DoD lands. And we did publish a paper in 2018 in Herp Conservation and Biology, which is a nice summary of herp diversity on DoD land. So I would encourage you to check that out if you're interested. So snakes on DoD lands. There are more snake species on DoD lands than any other herpetofaunal group, and that includes frogs and toads, salamanders, lizards, turtles, and crocodilians. There are 131 snake species confirmed present on continental U.S. military lands, and that represents 85% of all snake species in the U.S. So DoD lands are really important to the biodiversity of reptiles and amphibians, including snakes. Next slide. So we've documented 27 species of venomous snakes on military lands. And that becomes important from a safety standpoint, particularly when we have military troops training on the ground. And approximately half of the military sites with integrated natural resources management plans have at least one venomous snake species confirmed present. So integrated natural resources management plans are developed for military sites that have significant natural resources. And in total, there's about 340 military sites that had that requirement. So on about half of those, there's at least one venomous snake species confirmed present. Next slide. Probably without surprise, the venomous copperhead is found on more military sites, 84, than any other venomous snake species. And that's probably because the eastern copperhead is just really widely distributed across the eastern and central US. The most common non-venomous snake on military lands is the North American racer, and that occurs on approximately 200 military sites. Next slide. So DoD land support a few Endangered Species Act listed and at-risk species. We have the Eastern indigo snake, Eastern Massasauga, brown garter snake, and Louisiana pine snake, and the black pine snake are all confirmed present on DoD lands and are protected by the Endangered Species Act. And then also at-risk species include the desert Massasauga, the eastern diamondback rattlesnake, and the Florida pine snake. And both the eastern diamondback rattlesnake and the Florida pine snake are considered Department of Defense mission sensitive species. And I believe that's my last slide, and I will be kicking it over to Rob, who's going to be talking about our study. Rob?
Thanks, Chris, and thanks, Matt. And good morning, everybody. I wanted to start off by saying thank you to everybody from what Matt showed, what we know globally about snake fungal disease. And it's a huge community effort. There's so many people involved, and this project is emblematic of the same because there were so many people who were willing to participate within the DoD community and even outside with our partners and stakeholders abroad. So thanks to everybody, and let me roll the slides. Next slide. So the goals and objectives were to continue what we had started in 2018 and get a better feel. As Chris showed, there are a lot of bases, there are a lot of snakes, and there are a lot of questions surrounding what could happen with snake fungal disease going forward and how that could impact mission. And so there was a little bit of a COVID blip, and it extended our sampling time frame there in 21 and 22. Access to some installations was difficult early on, and so we adapted and got the work done, and these will be the results. So again, understanding the spatial distribution, what species are affected, do we have a lot of them? Are they protected? Are they not? Or none at all? And the environmental conditions under which we're seeing snake fungal disease or not. And then also, it's really important to get folks out there to learn more about snakes on their base. In a lot of cases, each of us as biologists for the military, we may be more knowledgeable on birds or fishes or whatever. And so it gives folks an excuse to learn more about maybe a group of taxa that they're not as knowledgeable about on their bases, et cetera, and then preventing the negative impacts to the military mission. Next slide. So we sent out a standardized field data sheet, a sampling protocol, and biosecurity procedures for how to clean stuff going from one site to the next so we don't transmit disease, snake fungal disease, and other, going from site to site within and among installations. We had a training video on how to swab, and that's on the internet. And so those still are out there for anybody carrying this forward beyond what we've done. We provided the kits for swabbing and then sent the results to Matt and his folks at the University of Illinois, and then provided a summary report. And I will say props to Matt, because it's not just a summary report, but his lab sent out individual letters to each installation that participated, showing them the results. And that created a back and forth of information and rapport, and helped us understand there were a couple errors on things that we cleaned up in the data set. And so again, that personal touch and reaching out to everybody involved was really important, and making sure we had solid data and solid understanding of what was found. Next slide. Here's some pictures just of the swab sampling, snake stretched out, everybody's gloved up, holding it, running the swabs up and down the snake, up and down, all around many times. And then you can see in the lower slide some examples of lesions that were detected and were reported to us in various species through photographs, beyond just writing it down on the data sheet and marking where they saw something. We got a look at what they actually saw with the photo submissions. Next slide. 47 installations participated in this, which is again tremendous. We appreciate everybody's assistance on that. 1,900 swabs from 826 individuals with 81 species or subspecies in 31 different states were observed and tested, and 36 of those species were detected with snake fungal disease. Next slide. I've got all my PPE on in that one picture there, clearly. Kind of like Jackass, don't submit videos trying to pull it off. Matt showed a slide earlier of the Baker et al. Categories, whereby we observed the conditions through qPCR and or were there clinical signs. And so I'm not going to belabor this slide, but these are the categories that we looked at for negative, present, possible, or apparent infection. Next slide. Nerodia had almost 3 times greater odds of being detected with snake fungal disease than other species. Interesting. It's aquatic, spends a lot of its time in and around water. Maybe life history, habitat use, having something to do with it there. And then dry marshon and colubra constrictor were more likely to exhibit signs externally if they were positive. Nerodia, again, were less likely to exhibit signs when positive, even though they had 3 times greater odds of being detected with snake fungal disease. And we got the first detections in the second study, the one we're talking about now, in New Mexico. And then previously, we got the first detections in Idaho, Oklahoma, and Puerto Rico. And that's huge. We wouldn't have that advancement in science and our understanding if DoD wasn't kind enough to fund this project, and we didn't all show up diligent to figure out where this thing is in the United States. So we're helping out biodiversity and science at large. Next slide. So here's the spatial distribution and what was sampled. The white states we did not get to. Some of those were sampled, or sorry, this is 2018, but still the white states were not sampled then. Light gray, no detection, but the dark ones is where we found snake fungal disease. So you can see a heavy prevalence to the right side, if you will, to the eastern United States, and then the 3 states to the left. And then in 2021, you see again, the white states not sampled. Some of those we had hit in 2018. Blue, no detection, and orange states detected. And fairly similar were western states are less represented, which happen to be drier states. And again, be careful of any correlations, but the eastern United States had much greater detection. Next slide. There we go. In summary, we had a greater prevalence in detection than the 2018 study. And I'll let Matt chime in when I'm all done, but I believe that's tracking with what we're seeing across the country from prior conversations, where we're almost at 20%. So 1 in 5 snakes that you pick up in the United States have the potential to have snake fungal disease versus 17% in 2018, at least on military lands. It's widespread on the landscape, on military lands. All 36 species had previously been documented in other studies with snake fungal disease. The high prevalence species have a consistently high prevalence throughout the literature, and the causes of that are still unknown directly. We got first positive in New Mexico, and we should all be aware to keep an eye on our snake populations on military lands so that those declines do not result in a pinch on the military mission through further necessary protections. What can we do going forward? Document, be aware, and report it back and report it up. Next slide. My pause might be because the computer's going slow on my end. There we go. Now I see the last slide. And with that, we will be happy to take any questions. That was a lot of ground we covered in pretty good time. It's only half an hour, so we do have time for some dialogue, and I'd encourage folks to do so. We've got 53 folks online, and thank you once again for joining us.
All right, so we're happy to take some questions. Charlotte's going to put them in the chat. If you have any questions for us about the project or about snake fungal disease, we're happy to take a stab at them.
And I'll say that anybody can reach out to Chris and I anytime, and we're available, and most folks know how to reach us. But we rarely have Matt, and he's been a heck of a partner in this endeavor for a number of years and a great colleague. And so make sure you guys ask questions of Matt while we have him. He's a busy guy.
Happy to be here. It's been a good collaboration.
Right on.
Yeah, and while we're waiting for questions, I'd like to thank the DoD Legacy Program for their funding of these projects. Without their funding, we could have never pulled this off. So thank you very much.
Yeah, good call, Chris. They're genuinely interested in supporting this, and that's fantastic.
All right, it looks like questions are coming through, and unless I'm told otherwise, I'll go ahead and read them and then answer them in the order that they come, if that sounds all right.
Okay.
Do it.
All right, so got a question about qPCR. What was the cycle threshold considered to be positive samples? So we run a complete 7 dilution standard curve on every plate, just to account for those minute variations on every plate. So I set the cycle threshold based on the lowest detection of the positive control dilutions, which will make sense if you're running those qPCRs, but typically it is around 33 is the cycle threshold, but you can obviously fluctuate that just artificially. So I don't know that 33 means a whole lot, but every plate went down to 10 to the 0 copies, so 1 copy. And if that dilution didn't amplify on a plate, it got rerun. That sample, every sample on that plate got rerun. The slides are going to be the slides aren't going to be made available, but that I'm aware of. But the presentation is being recorded, and the instructions on that were given out. But I'm guessing they'll be emailed as well. All right. Somebody had a question about the terbinafine, the treatment, terbinafine nebulization. Are you nebulizing dissolved tablets? Any concern for aerosolized or aspiration of particulate matter? Great questions. So tablets are crushed and dissolved in sterile saline into a solution that's put in the nebulizer, and it has a particular aerosolized particle size and these are human nebulizers that we've gotten from any distributor that does medical grade nebulization. We haven't had any concern about aspiration, but these are small particles and it's a mist. So there really isn't any solid particulate matter that's nebulized at all. Went through the presentation. Again, it's being recorded and will be available for you. And then there's somebody that had a more specific question about New Mexico. I'm copying down your email address, Clint, and Rob, Chris, or I will kind of coordinate those. All the results went through the contacts at the DoD, and those individuals have those results, and how to share those. We'll get back to you. Then a really great question about, is there plans to establish a frequency for which this study or surveillance will be repeated on DoD in the future? And I'd say we've had informal conversations about continuing or expanding or seeing where this surveillance goes. And, I think just like everybody, we're feeling like, which fire are we going to put out first? And with turtle shell fungal disease now somewhat taking a priority, especially with Western pond turtles, we're seeing how we're going to attack, to be honest, a lot of these herp-related diseases, because the goals of these surveillance in general, just across the board, is let's identify threats to conservation, but not all pathogens are threats. And to identify whether or not they're a threat or not, we have to have this information on surveillance on where it is and what's going on. I can tell you that certainly that snake fungal disease and Ophidiomices is a conservation threat to certain species, but not to every species. This DoD project was the first, and there's been a lot of snake fungal disease projects, but this was the first one to identify that Nerodia are actually carriers and less frequently associated with clinical signs. And that's an incredibly important piece of information for surveillance and understanding. Obviously, Nerodia are native in many areas of North America, and then they're non-native in certain areas in the West. And if we know that those are a species that is a potential reservoir or carrier that are unlikely to die from this, that's incredibly important information. So I would say that for all these other diseases, we're really in those, we need that baseline information. And because there's a limit on time and energy, trying to knock out some of those studies on baseline surveillance of Parananasiopsis or Mitomyces might take precedence over repeating the surveillance of snake fungal disease. Having said that, Rob and Chris and I have talked, and that if there are specific concerns at specific bases, we're wanting to find ways in order to test animals and species at your bases. And I'll let Rob or Chris chime in on those, but especially if you have individual concerns at your base, or your base has never been detected with it, but now you're starting to see signs, those are priority samples that I think we would find a way to be able to test. But you need to reach out to Rob, Chris, and I individually, kind of offline on those.
Yeah, Chris, I think it's fair to say you and I have a hair trigger on response to this, and it may sound a little trite, but a lot of what Chris and I do is look for vulnerabilities to the mission through the lens of reptiles and amphibians. And here we've done thorough studies on this disease across the DoD landscape and palette of landscapes, if you will, and we've learned a great deal. But as Matt was just saying and bringing it home, it's endemic to the environment, and it has localized effects, which can be disastrous to individuals and populations, but we're not seeing that in all taxa, and we have a lot still to learn. And so I think we'll have a phased approach to the next response as far as monitoring or any sort of DoD support for the disease. But right now, I think we're still catching our breath. But if you see specific problems, by all means, document them, bring them forward, And we have talked internally about setting up some mechanism whereby samples, we could have like an on-call sampling over a longer time period with just enough funding maybe to support if people see an incidence of a lot of detections or snakes are dying from it. We're sensitive to that, but we have not solicited further funding from DoD for anything yet. Still kind of huddling on all of that. Chris?
Yeah, I agree with everything you said, Rob. We're still trying to catch our breath on, finishing up this study and starting to look into the future. I could potentially see some additional testing in certain situations, particularly where maybe it was focused on, ESA listed species or at-risk species, or like you said, there was an outbreak was noticed. At a particular site. But as far as doing a very large scale, across the continental US sort of study, I don't know. I don't think that'll occur for a couple of years.
Yeah, and again, for everybody, listen up now. It's endemic and it's not an epidemic, but it can have disastrous effects on localized populations, individuals, and potentially species. We did not find that that's the case on DoD lands, but those things are happening in the United States to snakes like Massasauga, like the Lake Erie water snake. There's been some out there that we really have had problems with, and that's very similar to what Chris and I did a long time ago with so many others with the amphibian monitoring, when BD, an amphibian disease, was having really terrible effects and wiping out entire species throughout Central America and South America. And what we found was very similar to this, that it's endemic to the United States. It's having localized impacts like on mountain yellow-legged frogs, which had really bad impacts on, but not really the same for any other species, even though we found it in a lot of frog species. And so we had a data gap. We had a question: how is this disease operating? Is it as bad on our bases as we've seen in some other areas? And we're very fortunate that it is not, for what we know through these two studies, three, if you will, part three. I don't know if you guys have any more feedback on that, but
No, I think that covered it. We've gotten a couple more questions in.
yeah. Hey,
related
Matt,
to this.
real quick, the next one, Sabrina Kennedy.
Yeah.
So there's a gal, Raquel Elander, Elander. She works for California Department of Fish and Wildlife, and they were doing, and I think they're still doing, a huge study on it, snake fungal disease in the state of California. I would encourage Sabrina to, if you want to email me or Chris, or just write down Raquel Elander. I've got her contact info. Yeah.
She just took a new position
Oh.
within California Fish and Wildlife. I would say to reach out. We're still doing that project. We're running that with them.
Oh, cool.
And Deanna Clifford is the vet. And while DoD did not get any positives from bases in California, there are positives in California. The first one was published, so that one is out in the literature already, Sabrina. So you should be able to have access to that. But that study that Rob was just mentioning with that Raquel was the field tech that was coordinating it through CDFW. But Deanna Clifford is the PI on that project. It's a SWIG grant.
Well, shame on me, man. I forgot you were the conduit for all those samples. So I should have let you do that one anyway. Good times.
No worries. No worries. It's hard to keep track of this. I believe you.
Right on.
So, anything else, or do you want me to get back to some of the questions?
Yeah, go for it.
So I had a question about how long to keep samples for, and that's a good question. In the -20, a regular freezer that doesn't thaw, probably up to 6 to 9 months, and in the -80, there's no timeline on that. So collecting, if you have opportunity to collect samples and to throw them in a freezer, then definitely throw them in a freezer. Even if they're in the -20 for 5 years, we'd still run them, but the detection goes way down over time. We're just not sure how much it goes down over time. So yeah, collecting opportunistically, especially on snakes that look funky, I would, and put them in the freezer. There are still some states that were never sampled during the 2 DoD studies. Arizona being one of them. Some of the other states, North Dakota, really just have limited diversity of some snakes, so or have limited distribution of bases. So there are a few states that weren't sampled. I don't think we had anything from Washington either. So. Next question was about colony growth, too numerous to count on commonly used broadcast antifungals that I mentioned, and about mentioning SFD increasing after the product entered the market. The product was an azole antifungal, which we use azole antifungals to treat reptiles, amphibians, mammals, birds for lots of fungal diseases. Not sure what it means about its emergence, because we found ophidiomyces, and the National Wildlife Health Center have found ophidiomyces in museum specimens dating back to the 1930s, but not necessarily associated with disease. I think most biologists, you know, that were working in these times knew about hibernation sores for sure. But really in the late 1990s and early 2000s, there was some switch in the clinical signs and the physicals exam findings from some of these animals. And it's definitely an area for future research on why there's been a change in the previous 20 years. And so whether or not it has anything to do with our altered land use patterns and procedures, I think is definitely possible. But I think that's an area for future growth. There's no cause and effects that have been established. Colin corrected me. Washington indeed had. So then maybe it was Oregon. Although I'm probably wrong on that one too. There were the first 2 studies had 62 bases, and this one had nearly 50. So it was a lot of different bases.
Yep.
Yeah, we did sample in Oregon as well.
Okay, thanks, Chris.
Yep.
Last question, I think probably before we need to probably call it is, were there any trends regarding physical signs of infection on vipers? Looking specifically on eastern diamondbacks, no, there are not. Vipers, rattlesnakes in particular, are uniquely sensitive to ophidiomyces across the board. The least affected viper that we know of is cottonmouths, but and copperheads, which are the most common, as Chris said, on bases, really show minimal clinical signs. But diamondbacks in particular, timbers, Massasaugas, all are uniquely sensitive and get lots of clinical signs. They look pretty nasty, but there's been no progression regarding or trends. They get all 5 lesion types, and they usually get them worse than most other non-venomous species.
Thanks, Matt. I did want to add to the group before we cut out that Matt mentioned the Western pond turtle and turtle shell disease, and that truly is gaining momentum, even on Chris and I's radar. And so Matt is running samples for our range-wide pond turtle project that we just finished year 1 on installations. The only installations left where there's pond turtles are in California, and we hit up 11 of them this year. And so we'll be coming back in year 2 and gathering more samples. I don't think we have any results on that, but turtle shell disease is prominent, and that's kind of our gaining prominence, unfortunately. But that's our starter set, if you will, within DoD for Chris and I to begin to evaluate turtles and what's going on with them. As that disease again takes greater, it's just becoming a little bit bigger. I don't know if you want to say anything about that real quick, Matt, but you'll be hearing more from us on that as we go forward, and hopefully we'll have a webinar a year or so from now on that topic.
Yeah, Amidomyces is a major player in Western pond turtles, and we're in Blanding's turtles. Um, and so we're seeing it in red-eared sliders. We're seeing it all over. So, but not in the wild except for Western pond turtles. And this is what makes it different from Ophidiomyces. We just finished a 1,400 turtle surveillance effort of 7 different species in Illinois, and found it in 6 animals that were kind of in an artificial pond, prevalence of less than 0.2%. So this is where it's different than Ophidiomyces, which it's endemic across the entire range for the most part of North America. Amidomyces is not there yet, and especially not in the Midwest. And with all the problems that the Western pond turtle are having, conservation problems with this, we don't want it in the Midwest. And so what it's been associated with are Head Start programs. And that's where we identified 35% prevalence in one of our Head Start Blandings groups. And we were able to catch it before those animals were released. And those animals weren't necessarily showing signs. Those were surveillance samples. So this is definitely the area that's kind of taken over my life a little bit recently. But I feel like, as opposed to reactionary medicine and procedures that we're having to do with Ophidiomyces, in this case, it can be much more proactive with Amidomyces in much of the range.
Matt, we got one more last question from Anilis, our colleague in Puerto Rico. And not to paraphrase, but do we see the same trends, like the perfect conditions for BD in cloud forest environments, like 60 degrees or whatever, with that high humidity? Are we seeing any climatic conditions that really favor it? And that's a great question.
It is, and that's a much deeper dive, because we didn't get temperatures at the time of collection for any of the DoD project, and so it's a dive into NOAA data to try to identify what were mean temperatures at each of these bases at each of these times of collection. So that's a little bit beyond. It was deemed beyond the scope for getting these reports out, but it definitely is good for future evaluations. I will tell you, it because of the delay in from exposure to detection in many of these cases, that I don't think we're going to see environmental temperatures play a role. It's not immediate, meaning that if 85 degrees Fahrenheit is the key, point of infection, and that happens in July, but it takes 9 months for it to develop, and we test them in March of the following year, then it's not associated with the detection in March. It's obviously associated with when they were exposed in July, which we have no idea of when they were exposed, because these guys can show signs anywhere from 2 months to 2 years after exposure. So I don't think that data is ever going to be clear. I think we identified it from experimental transmission to know what the factors are, but I don't think. I mean, BD happens on a much shorter time frame, even though it's still somewhat prolonged. I just don't think we're going to ever get that data on Ophidiomyces.
I will add, Matt, at least from personal experience, and Chris was there, but when we were sampling, I think it was 2018 in Pennsylvania, and that was a year that they had an exceptionally late, wet, relatively warm, a lot of rain, warm spring, and the rain stopped. Like, we were there for meetings and then field work. The rain stopped the day of our last meeting when we were hitting the field. And when we went out, the sun came out, and we were just thankful for the great conditions, because we wanted to find stuff, and it was raining, not just, aquatic stuff that likes rain. And we found a ton of stuff. It's like every snake and their brother came out and started to enjoy that sun on the day we were there. And every snake we found among many species on that day, it was an epic day. We caught timber rattlesnakes, water snakes, hognose snakes, rat snakes, every single one, I think, tested positive for snake fungal disease and had ulcers and lesions on them. That somehow, again, maybe a false correlation, but all that wet definitely didn't benefit them. And that was when my eyes kind of opened, like, wow, I've never seen anything like this. When you get this many snakes, you have such a great day catching them and seeing them, and every single one had snake fungal disease, and we provided swabs. So be on the lookout.
Great. Thanks, Rob. Thanks, Chris.
I think
Thank
we're
you.
good.
Everybody participating and watching the seminar, and look forward to hearing from you if you have any questions.
Yep.
Yep, thanks again, Chris, Rob, and Matt. Thanks everyone for coming, and thank your leadership for giving me the hours to attend today. And we look forward to seeing everyone next time.
Great.
Take care,
Wonderful.
everyone.
Thank you. Goodbye.
Bye.